Okay, so ladies and gentlemen, you are all welcome to my YouTube channel. Today we are going to look at the concept of transgenicity. What is transgenicity? Definition of transgenicity. Transgenicity refers to the process of introducing genes from one species into another to create genetically modified organisms, that is GMOs. So this involves genetics engineering techniques to alter the genetic makeup of an organism. So it means that in transgenicity, what actually happens is when you get a gene or you transfer a gene from from one organism. Let's say, for example, there is a gene A in let's say a bacteria. There's a gene A in bacteria, and you now transfer that gene from bacteria into another species of organisms. So that process is called transgenicity. And the organisms the organism that received that gene from species of any other organisms is called genetically modified organisms because the genome of that organism is altered. So as a result of that alteration, then the organisms is called genetically modified organisms. And this process of transgenicity usually involves the genetics engineering techniques that used to alter the genetic makeup of an organism. That is transgenicity. It's a process that involves the transfer of gene from organisms A to another organisms or from one species of organisms to another species of organisms, and that is what described as transgenicity. And then how this transgenicity works. So transgenicity works by isolating the desired gene of one organism, inserting it into the DNA of another organisms, and allowing the recipient organisms to express the gene. And this results in the transfer of specific traits or transfer of character. Like, for example, this is a typical example. There is a one bacteria called Bacillus thuringiensis, that is BT organisms, and they are known that these organisms, they have insecticidal protein. Insecticidal, they have insecticidal, they have insecticidal protein in the bacteria. And of course, we know that a lot of microorganisms or a lot of insects are destroying our crops. So what scientists did is actually isolating this insecticidal gene from the VT bacteria, and then introducing that gene into the another organisms. And the another organism that this insecticidal gene introduce is, a typical example is cowpea. And as a result of that introduction of the cowpea now become insecticidal to any insect that come close to them. So you see, as a result of that, insecticidal gene is isolated from BT bacteria, then is now introduced into the cowpea, and as a result of that, a new there is a transfer of a specific trait or a characters, and that specific trait or character in the cowpea is the insecticidal property. So any insect that feed on the cowpea due to the presence of this insecticidal protein, then the insects will die. They will die. So as a result of that, that is how the transgenicity works. You transfer a gene or you isolate a desired gene from one organism, then you insert the gene into another organism, and the recipient organisms will now express that gene, and then that result in the transfer of a specific character. So this is how transgenicity work. And then the examples of transgenicity is like common example of transgenicity include genetically modified crops where a gene of traits like pest resistance or increased yield are introduced. So other examples include transgenic animals used in research or gene therapy. So now, what are the benefits and the challenges of transgenicity? Challenges of transgenicity. On the flip side, transgenicity raises concern about environmental impact, biodiversity, risks, and ethical consideration related to genetic manipulation. And there are also debate about the food safety and a long-term effect in ecosystem. And this is how typically some of how these genes are introduced. Sometimes you use a viral vector to introduce the gene uh, into the new organisms. Like for example, this is an organism. So this is an injection. You get the gene, you introduce the gene into the viral and you inject the viral into human, like for example here, then of course the gene will be ejected into the human. So this is how it happens. This is how it works. And then what are the benefits? So the advantage of the transgenicity, it has benefits such as it improves crop yields, and then it also enhances nutritional content, resistance to pests or disease. It also enables the production of valuable proteins like insulin through genetics modification. So these are the benefits and the challenges of transgenicity, and this is how it's exactly work. Like for example, you isolate the cells from human. After you have the cells, you culture the cells in vitro, and then you introduce the gene into the cell. After introducing the cell into the cell, then you now transfer that gene back into the humans. So this is how example of gene therapy is applied. So it means that here you get a gene and you introduce it into the vector, and then you now introduce into the viral vector, and then you now use the viral vector and introduce the gene into the cell. So after the gene is introduced into the cell, then it now transfer back into the human. And this is the concept of gene therapy. So thank you for listening. And if this is the first time you are coming to my YouTube channel, please subscribe.